Hello everyone. Today we're going to learn about cytokines. Cytokines are very important. So let's start learning. The development of an effective immune response involves lymphoid cells, inflammatory cells, and hematopoietic cells. The complex interactions among these cells are mediated by a group of proteins collectively designated cytokines to denote their role in cell-to-cell communication. Cytokines are low molecular weight regulatory proteins or glycoproteins secreted by white blood cells and various other cells in the body in response to a number of stimuli. These proteins assist in regulating the development of immune effector cells, and some cytokines possesses direct effector functions of their own. This chapter focuses on the biological activity of cytokines, the structure of cytokines and their receptors, signal transduction by cytokine receptors, and the role of cytokine abnormalities in the pathogenesis of certain diseases and therapeutic uses of cytokines or their receptors. The important role of cytokines in the inflammatory responses dealt later. So we won't be dealing with that today. Okay. So we're going to learn some aspects related to properties of cytokines, cytokine receptors, cytokine antagonists, cytokine secretion by Th1 and Th2 subsets, cytokine related diseases, therapeutic uses of cytokines and their receptors, then cytokines in hematopoiesis. But I will be excluding some portions related to clinical application, so pardon me for that. Then properties of cytokines. Cytokines bind to specific receptors on the membrane of target cells, triggering signal transduction pathways that ultimately alter gene expression in the target cells. The susceptibility of the target cell to a particular cytokine is determined by the presence of specific membrane receptors. In general, the cytokines and their receptors exhibit very high affinity for each other. This dissociation with dissociation constants ranging from 10 raised to minus 10 to 10 raised to minus 12 m. Because of their affinities are so high, cytokines can mediate biological effects at picomolar concentrations. So in those minute quantities they can act. So a particular cytokine may bind to receptors on the membrane of the same cell that secrete it, exerting autocrine action. It may bind to receptors on a target cell in close proximity to the producer cell, exerting paracrine action. In a few cases, it may bind to target cells in distant parts of the body, exerting endocrine action. So cytokines regulate the intensity and duration of the immune response by stimulating or inhibiting the activation, proliferation, and differentiation of various cells, and by regulating the secretion of antibodies or other cytokines. As described later, binding of a given cytokine to responsive target cells generally stimulates increased expression of cytokine receptors and secretion of other cytokines, which affect other target cells in turn. Thus, the cytokine secreted by even a small number of lymphocytes activated by antigens can influence the activity of numerous cells involved in the immune response. For example, cytokines produced by activated Th cells can influence the activity of B cells. TC cells, natural killer cells, macrophages, granulocytes, and hematopoietic stem cells, thereby activating an entire network of interacting cells. So, cytokines exhibit the attributes of pleiotropy, redundancy, synergy, antagonism, and cascade induction, which permit them to regulate cellular activity in a coordinated, interactive way. And each of these topics will be explained later, so don't worry. A given cytokine that has different biological effects on different target cells has a pleiotropic action. So that is what is pleiotropic. A given cytokine that has different biological effects on different target cells has a pleiotropic action. Two or more cytokines that mediate similar functions are said to be redundant. Redundancy makes it difficult to ascribe a particular activity to a single cytokine. Cytokine synergism occurs when the combined effect of two cytokines on cellular activities is greater than the additive effects of the individual cytokines. In some cases, cytokines exhibit antagonism, that is, the effects of one cytokine inhibit or offset the effects of another cytokine. Cascade induction occurs when the action of one cytokine on a target cell induces that cell to produce one or more other cytokines, which in turn may induce other target cells to produce other cytokines. Here we could see some pictures. Overview of the induction and function of cytokines. Most cytokines exhibit autocrine and paracrine action. Few exhibit endocrine action. Here we could see a cell, which is a cytokine producing cell. Actually, a cytokine producing cell. There must be some inducing stimulus. So the cytokine gene inside of that particular cytokine producing cell got activated. Later that will be producing cytokine, which travels and binds to the receptor on the target cell, which induce some sort of signals. Then gene activation will be taking place inside that target cell, which will be eventually leading to biological effects. So just try to draw this picture if you are undertaking a written examination. So inducing stimulus, cytokine gene, cytokine producing cell is that with its own cytokine gene, and inducing stimulus will be approaching that particular cytokine producing cell, then that will be leading to the production of cytokine. Later another target cell will be there, which has some sort of gene activation happening because of the signal produced when the cytokine bind to its receptor. Then gene activation will be taking place, which will eventually lead to biological effects. And along with that, some other modes of action are also shown. Here you could see autocrine action. Autocrine in the sense cytokine is produced from cell A and the same cytokines are acting on the receptors of this cell A. But in case of the paracrine action, cell A is producing cytokines, but the receptors are present on the nearby cell. So there will be some sort of traveling happening, not some sort of elaborate traveling, just neighboring cells will be getting activated because of this cytokines produced by some cytokine producing cells or the neighborhood. Then endocrine action, some source cells or organ produce these cytokines. Then with the help of blood or lymph, they travel into distant places. So distant cell act as a receptor. Okay. So these are some sort of mechanisms with the help of pictorial depiction. So if you are undertaking a written examination, I prefer to draw these pictures. Okay. Then next we'll be learning about the term cytokine. Encompasses those cytokines secreted by lymphocytes, substances formerly known as lymphokines, and those secreted by monocytes and macrophages, substances formerly known as monokines. Although these other two terms continue to be used, they are misleading because secretion of many lymphokines and monokines is not limited to lymphocytes and monocytes, as these terms imply, but extends to a broad spectrum of cells and types. For this reason, the more inclusive term cytokine is preferred. Many cytokines are referred to as interleukins, a name indicating that they are secreted by some leukocytes and act upon other leukocytes. So interleukins 1 to 25 have been identified. There is no reason to suppose that still other cytokines will be discovered and that the interleukin group will expand further. Some cytokines are known by common names, including the interferons and tumor necrosis factors. Recently gaining prominence is yet another subgroup of cytokine, the chemokines, a group of low molecular weight cytokines that affect chemotaxis and other aspects of leukocyte behavior. These molecules play an important role in the inflammatory response and are described later. So, because of cytokines share many properties with hormone and growth factors, the distinction between these three classes of mediators is often blurred. All three are secreted soluble factors that elicit their biological effects at picomolar concentrations by binding to receptors on target cells. Growth factors tend to be produced constitutively, whereas cytokines and hormones are secreted in response to discrete stimuli, and secretion is short lived, generally ranging from a few hours to a few days. Unlike hormones, which generally act long range in an endocrine fashion, most cytokines act over a short distance in an autocrine or paracrine fashion. In addition, most hormones are produced by specialized glands and tend to have a unique action or one or a few types of target cell. In contrast, cytokines are often produced by and bind to a variety of cells. The activity of cytokines was first recognized in the mid 1960s when supernatants derived from in vitro cultures of lymphocytes were found to contain factors that could regulate proliferation, differentiation, and maturation of allogenic immune system cells. Soon after, it was discovered that production of these factors by cultured lymphocytes was induced by activation with antigen or with non-specific mitogens. So biochemical isolation and purification of cytokines was hampered because of their low concentration in culture supernatants and the absence of well-defined assay systems for individual cytokines. Great advance was made with the development of gene cloning techniques during the 1970s and 1980s, which made it possible to produce pure cytokines by expressing the protein from cloned genes. So the discovery of cell lines whose growth dependent on the presence of particular cytokine provided researchers with the first simple assay systems. The derivation of monoclonal antibodies specific for each of the more important cytokines has made it possible to develop rapid quantitative immunoassays for each of them. Now we're gonna look into some pictures, so carefully view this pictures. These are some pictures which describe cytokine attributes of pleiotropy, redundancy, synergy or synergism, antagonism, and cascade induction. Okay, so this first picture which depicts pleiotropy. We mentioned what is pleiotropy. I hope you know that. If you don't, then no problem. We're gonna read that once again. A given cytokine that has different biological effects on different target cells has a pleiotropic action. So one cytokine is there with different biological actions or different biological effects on different target cells. Okay, that is called as pleiotropic action. So here we could see a activated TH cell. Activated TH cell is there. It shows the property of pleiotropy. Pleiotropy showing in the sense cytokines show that. Okay, pleiotropy nature. But we'll be describing like that. So IL-4 is secreted, which is actually a cytokine. So which act on this first B cell, which is acting as a target cell and producing some effects related to activation, proliferation, and differentiation of this particular target B cell. Once again, I'm going to explain this. Activated T cell is there, which produce IL-4 named cytokine, which produces some sort of effect in this particular B cell, which is acting as a target cell. What are those particular effects activation, proliferation, and differentiation related to this B cell, which is acting as a target cell. Okay. Then next, a thymocyte is also there, which is acting as a target cell. The same IL-4 is present, which is the cytokine, which acts on the receptors of this thymocyte, which leads to the proliferation of that particular cell. Okay. Then some sorts of other cells called mast cells are there. With the help of this IL-4 cytokine, that undergo proliferation. Okay. So I hope this point is clear. Activated pleiotropy, we are mentioning about the topic called pleiotropy of the cytokine. IL-4 is there, which is released from activated Th cells. First, in this picture, you could see it's acting on the receptors of this B cell, which undergo some sort of changes, which will eventually lead to activation or proliferation or differentiation of this B cell. Then IL-4 is acting on thymocyte, which will eventually lead to the proliferation of this thymocyte. Same is the case with mast cell, but there will be proliferation happening. So this whole scenario explains pleiotropy. Then on the left side, we could see another picture which shows cascade induction. Activated Th cell is there, which releases IFN gamma, interferon gamma, I guess. I'm not sure what the full form, so just learn it like IFN gamma. I mentioned I am a beginner, so don't judge me. IFN gamma, macrophage is dead. This activated Th cell secrete IFN gamma, which is the cytokine. Okay, so that is going to be a very important part. So IFN gamma is a cytokine which acts on this macrophage later, which will produce IL-12, which is another cytokine. Then that IL-12 will activate another Th cell, and activation lead to some sort of reactions which secrete some other important cytokines like IFN gamma, TNF, tumor necrosis factor, IL-2, and other cytokines. Okay, so this is what happens in case of cascade induction. Once again, I'm gonna shortly describe this. Activated Th cell is there, which produce IFN gamma, which will eventually activate macrophage, which lead to the secretion of IL-12 cytokine. Then activated Th cell, another one, then that will produce IFN gamma, TNF, IL-2, and other cytokines. Okay. Yeah. The next point was redundancy. Redundancy in the sense activated Th cell is there, which produces different cytokines, IL-2, IL-4, and IL-5, which acts on B cell proliferation. Okay, redundancy. Even though these cytokines are different, the action or the biological effect which they produce is same. That is what we mentioned with the help of this picture of this redundancy of cytokines. Then synergy, activated Th cell is there. IL-4 and IL-5 got released from this activated Th cell, and they cumulatively, exert some effects on this B cell, which induces class switch to IgE. Okay. Then antagonism, activated Th cell is there, and IL-4 is got released from this activated Th cell. Then if that IL-4 is present, there must be some sort of class switch, as we mentioned earlier. Okay, but IFN gamma is another cytokine which is also released from this activated Th cell, that will eventually block this binding of IL-4 to its receptors on B cell. So there must be some sort of mechanism which blocks class switch to IgE induced by IL-4. Okay, that is what is explained with the help of this picture which depicts antagonism. Okay, so we mentioned some important attributes of cytokines like pleiotropy, redundancy, synergism, antagonism, and cascade induction. Okay, then next we need to learn something about the practical application of this topic. Practical application in the sense laboratory application. So some experiments were conducted, and I'm not sure about the explanation of this point, but still I'm gonna try this. So this figure depicts ELISA assay of a cytokine. Okay, so this particular sample, which is depicted here, pictorial depiction with portions like cytokine bound to antibody coated onto well add enzyme conjugated secondary antibody that later that will lead to add substrate and measure color. There must be some color change. Yeah, let's read this. ELISA assay of a cytokine. The sample containing the cytokine of interest is captured by specific antibody. That particular antibody is represented with this blue color. Okay. Then coated onto wells of a microtiter plate. A second specific antibody conjugated to an enzyme E, such as the horseradish peroxidase, forms a sandwich with the captured cytokine, immobilizing the enzyme in the microtiter well. A chromogenic substance S is added, and the enzyme generates a color whose intensity is proportional to the amount of cytokine bound to the capture antibody. The optical density of this color produced by the unknown is compared with values on an appropriately determined standard curve. The standard curve shown here is for human interleukin 12, IL-12. It is clear that this assay is sufficiently sensitive to detect as little as 1 picogram of IL-12. Then this picture. Yeah, I'm finding it a bit difficult to explain this, so I won't be explaining this. Sorry. I'm just gonna read this. Okay. Several representations of structures in the hematopoietin family. Topographical representation of the primary structure of IL-2 showing alpha helical regions, alpha and AD, and interconnecting chains of the molecule. Proposed a three-dimensional model of IL-2, ribbon model of IL-4, reduced from X-ray crystallographic analysis of the molecule. And alpha helices are shown in red and beta helices in blue. The structure of other cytokines belong to the hematopoietin family are thought to be generally similar. So just look at this figure. Okay.